the Ren lab who's built a tool called CATlas. And what CATlas does is essentially allows you to visualize accessible chromatin regions using ataxic data. So I'll hand it over to you.
Okay, cool. Thank you. Let me share my screen. Okay, can everyone see my screen now?
Yes, looks good.
Okay. Okay, afternoon, everyone. This is Yang from BICCN's lab. Before I go to the web portal, I want to give you a brief introduction about the data set we have and what analysis we did in this data set. So we know we currently have achieved a great achievement to explore the cell type and annotate the cell type by using the single cell RNA-seq data. And by using this single cell RNA-seq data, we are able to annotate the cell types based on cell type specific expressions. And to understand how those genes are differentially expressed in different cell types, we use the taxic data, try to annotate the potential enhancer and promoters, and also try to understand how those cis-regular elements regulate the gene expressions in different cells. So 2 years ago, Sebastian from our lab have developed this single nuclear taxic technique, and by using a combinatorial barcoding strategy. And Xiaomeng from our lab generated all the single nuclear taxic data in this current project. And as Panqing just mentioned, in current data we have, we are sampling we are M-bytes sampling neurons and non-neurons from 45 brain dissections, and over 800,000 nucleus are included in this study. And to identify the cell clusters and annotate the cell types, we basically we identify the cluster in 3 levels. So the first level, we cluster the cell clusters into 3 groups. You can see the first group is the non-neurons. They are colored in purplish. In total, we have 250,000 nucleus are annotated as non-neuron cells. And you can see also we have identified 160,000 nucleus are annotated as a GABAergic neuron, and the rest of the nucleus are annotated as a GABAergic neuron. And for each of the major groups, we further get the clusters and annotate the clusters as a major type. For example, here, MGL means the microglia, and ASC represents the astrocyte. And to explore the subtypes for each of the major type, we extract the cell from the major type and perform another round of cluster in order to find the subtypes of this major type. I will show one example here is the SST GABAergic neuron. As you can see, we are able to identify 10 subtypes of this SST positive GABAergic neuron. And one of them have annotated as long projecting GABAergic neuron, which have a marker gene previously defined from RNA-seq data, is a gene called CHODO. You can see in this subtype, only in this subtype, you can see the sensibilities along the gene body of CHODO gene. We annotate those major type and subtypes previously based on the published marker genes. And we also did the integrative analysis with previous published single cell RNA-seq data. And by performing the integrative analysis, we can pretty well align the cell types found in this study to the previously defined taxonomies. And since we are dissect the brain to detail brain dissections that give us the power to explore the regional specificity of different brain cell types. As shown in the upper panel here, we actually showing the cell compositions for each major type and subtypes in different brain regions. And we also calculate the regional specificity score for every subtype. In the lower panel, I just show you one example here. Here I show few subtypes finding PV positive GABAergic neuron. And you can see one of the subtypes are mainly from the striatum region. We annotate these subtypes as striatal PV positive interneurons by checking one of the known marker genes. Okay, so after we annotate those cell types, we are trying to explore and identify the cis-regular elements in each subtype. We basically call the peaks from each pseudobug data in each subtypes. And then in total, we are able to identify 491,000 candidate cis-regular elements in current data. And by grouping them into different modules, we find, except the module 1, which have the cis-regular element mainly located at the promoter region, you can see the distal regions, majority of the distal regions show great cell type specificity. And to take advantage of those cis-regular elements we defined in each subtype, we are able to explore the different cis-regular elements in different subtypes, and also predict the potential driver TFs in each subtype. Here I show one example is the one of the subtype of SST positive GABAergic neuron, which is the cluster 10 here. We identify the differential cis-regular elements for this subtype. And using those differential cis-regular elements, we perform the motif analysis. And also we explore the expression level of this. From the motif analysis, we find there the TFs in KLF families are highly enriched in the subtype. And we also explore the previously published RNA-seq data. And we are thinking the KLF5 could be one of the novel factors in this subtype. So, another aspect to explore the data is actually very interesting in how the cis-regular elements are regulated the expression of different genes in subtype. So to predict the potential linkage between distal elements and the gene, we basically use two strategies. We use two steps to predict the linkage between distal elements and the gene. So the first step, we are identify the co-accessible cis-regular elements from the ataxic data. So if there are two regions are co-accessible, so it's more likely those two regions are physically close to each other in that subtype. And they are potentially, this distal elements are regulate the expression of this gene. And the second step, after we identify those co-accessible regions, we also calculate the Pearson correlation between the RNA expression and also the cis-regular element accessibilities. By comparing with background, we are able to identify 129,000 positive correlated pairs between the gene and the distal elements. By using those linkage we predicted between the gene and the distal elements, where we can use those information to explore the potential mechanism behind the cell type. For example, here I show 3 heat maps. The left one showing the accessibility scores for every distal element. And the heat map in the middle showing the expression level of its target gene. And the right panel showing the accessibilities as a promoter region. So take advantage of those linkage, we are able to use the cis-array elements to explore the potential TF in the cell types, and using the potential target genes to explore the potential biological process involved in this cell type. Since we are unbiased to sample the cells from the brain, which give us the power to explore several lineages. And for example, I'm showing here is the maturation of oligodendrocyte. We're able to explore the potential driver TF involved in this lineage. And also we're able to explore the 2 lineage in adult neurogenesis, and also predict the potential driver TF involved in these 2 lineages. Besides, since we are predicting the potential linkage between distal elements and the gene, so you can explore the data in more detail. For example, here I show you the neural intermediate progenitor cells came from 2 brain regions, or 2 neurogenesis zones, one from subventricular region, the other from subgranular zone. By exploring the data more deeply, we're able to link the potential regular elements we defined in this study to its potential regular genes. We also explored the whether those cis-array elements we defined are associated with potential neurological disease. And here I show you the GWAS analysis and show you the enrichment of each major type in neurological disease or other GWAS traits. Here I also show you one example. Here is the 28% of the potential causal variance in schizophrenia actually is overlap with the distal cis-array element we defined in this current data. As I can show you here, the 4 gray bar here showing the cis-array element we defined in this current study. And each dots here represent 1 potential causal SNPs involved in the schizophrenia. Okay, I will stop here and share you the web protocol we constructed. Okay. So this is our web protocol to visualize all the data set I just presented. Our protocol called CATlas, short of the cis element atlas. You can type catlas.org/mousebrain. Let me send it through the chat. Okay, in this protocol, we provide 3 major functions. So the first function is able to explore the cell clusters we defined in this current study. And the second function is we provide the detailed annotation information for each cell type. And the third functions, we provide a IGV browser, genome browser to explore the signals for each cell type. So for more details, if you are interested, you can go to the preprint on BioCAF. I would like to mention 2 softwares I used in this protocol. One is the USSC single cell browser, and the other is IGV JavaScripts developed by IGV teams. I use those 2 tools to visualize the current data we have. So you can click the cell browser at the bar here, or you can click the bottom export here. That will guide you to the first function we provided in this byproto. So as I mentioned, we define the cell clusters and annotate the cell types in different level. So we provide the cell embeddings of those nucleus in different level. For example, here we show all the non-neurons, the cell embeddings for all the non-neurons, which included 10 major types. Or if you're interested in the subtypes, you can go to here, you can click the open. Then they will show you the cell embeddings for each major type. And you are able to export the subtypes, the cell embeddings for the subtypes. For example, here I will show you the example of a subtype of astrocyte. You can click the open here. And here, just show you, we are able to define three subtypes from the major type astrocyte. And you can view the cell embeddings in different ways. First, you can split the screen into two. And as you can see from the left panel, showing there are multiple options for the cell annotations. And you can explore, for example, whether the cell clusters are influenced by the biological replicase, or the unique mapped fragments, how the unique mapped fragments are calculated for every cell type. They are unbiased, showing in different subtypes. And you can also explore the TSS enrichment for each cell type. Also, if you are interested in the major regions or subregions where those nucleus come from, you can click the annotations here. For example, I click the major regions here. Those cell types will colored by the major region. And you can also go to subregion. Okay, so in the second panel, you can explore the gene activity scores for some marker genes. For example, in this subtype of astrocyte, we find the previous annotated lineage called oligo2 lineage. So I can simply search the gene oligo2 here. Then they will show you the gene activity score for each nuclear for this gene. And if you are not satisfied with this color pattern, you can change the pattern. You can change the color here. For example, I choose this color pattern. Okay. There's another way to explore the data. For example, I'm interested in the cell type. I'm interested in the nucleus from pallidum. I can simply select the nucleus from pallidum here, and then you can explore the gene activity scores for the nucleus come from this brain region, comparing to the other nucleus. Okay. So another example I would like to show you is the SST GABAergic neuron, as I previously showed in the slides. You can also explore the gene activity scores for some marker gene. For example, I show you here is the gene activity score for one of the marker gene for long projecting GABAergic neuron, and you can use this tool to select the any nucleus you are interested to explore the gene activity scores, calculate for each nucleus. Okay. So then I will move to the, so if you're interested in explore more detail about each data set, we provide a download for For example, if you're interested in the subtype of astrocyte, we provide the download here, which included the cell by gene matrix and the meta table and the cell embeddings in UMAP and other informations. Okay, so the second function we provide in the portal is the cell type annotation. We provide a table here. You can use this table to explore any cell type you're interested in. For example, I was interested in the astrocyte. That will show you the 3 major astrocyte subtypes we find in this study, or you're more interested in progenitor cells. Progenitor cells. By clicking this button, you can view more information for the cell type. We included the QC controls and where those nucleus are coming from, different brain regions, and we provide an IGV track. You can use that to explore the open chromatin signals for the cell type. And for the third function we provided in this portal, we provide a IGV genome browser. You can use this IGV browser to explore the accessibility scores in each subtype. You can select any subtype you are interested in. For example, I will select the SST trotol cell here. Then they will load it to this view. And for each subtype, we have 3 tracks. The first track just shows you the accessibility open chromatin signals along the genome. The second track showing the linkage we predicted between a cis-rare element and a gene. And the third track showing the cis-rare elements we identified for the cell type. And you can click the setting buttons here to change, for example, the track head or the color or the name. You can also, for example, flip the direction of those arcs. Okay, let me show you a few examples. You can search either the genome coordinate or search by the marker genes. For example, I search the trotol genes. Then I can zoom in or zoom out by using these 2 buttons. For example, I can show you the SST trotol cell have more accessibility at the gene body of trotol compared with the other subtypes. And we identified 2 potential cis-rare elements are likely regulate the expression of this gene. And also, for example, we can explore other genes. For example, here is the one of the subtypes for astrocyte, which previously annotated as oligo-2 lineage. You can see the potential cis-rare elements are linked to this gene. Okay, so at last, we'll continue develop this web portal to provide more information. And please do not hesitate to contact us if you have any questions. Okay, that's it. Thank you.
Thank you. All right, Michelle, do you want to stop recording?